Carnegie scientists explore the outermost reaches of space, the mysterious depths of the Earth, and the origins and mechanisms of life. Our brilliant and dynamic teams of scientists transform the landscape of discovery with their unbridled passion, insatiable curiosity, and never-ending quest for knowledge. With limited teaching, administrative, and grant pressures, Carnegie's scientists concentrate on research and are empowered to go their own way. Our scientists are chosen for unique skills and boldness. Their amazing discoveries, spanning more than a century, demonstrate the power of freedom. Carnegie investigators are intrepid adventurers who pursue their goals even as they mentor and train the next generation of scientists. They are free to question, to wonder, to creatively pursue ideas from the vast universe to the subatomic world. Our scientists redefine the pursuit of what's possible for us, our planet, our universe. We explore the past to understand the present and inform the future.
Thank you, everyone. Before I introduce Rebecca, I will just say that just get some logistics out of the way. So I wanted to say that if anybody has a question for Dr. Albright, please place them in the Q&A chat before in the Zoom, and then at the end of the talk, we will ask Rebecca those questions for her to answer. And so it's with great pleasure that I introduce my friend and colleague, Dr. Rebecca Albright, for our seminar today. Dr. Albright is a curator at the California Academy of Sciences in the beautiful Golden Gate Park in San Francisco. She's actually a former postdoc at the Department of Global Ecology at Carnegie, which is on Stanford's campus. Dr. Albright is a coral reef biologist with dynamic expertise in biology, ecology, and biogeochemistry. Her research her career really has focused on understanding the ability of coral reefs to cope with changing environmental conditions. She has studied coral reef ecosystems all across the globe, including in the Florida Keys and on the Great Barrier Reef in Australia. Dr. Albright is particularly interested in understanding how changing global conditions caused by climate change will affect coral sexual reproduction and what this might mean for the next generation of corals as the world warms. At the Academy, Dr. Albright is currently developing methods to spawn corals in Golden Gate Park to try to develop new methods to maximize coral reproduction in the field and learn how to manage these awesome organisms. In addition to her commitment to research, Rebecca's passion for community engagement has been fostered by years of working with nonprofits and citizen science organizations that focuses on educating communities about coral reef conservation, their management, and just the basic fascinating biology of these organisms. Her work has been featured in prestigious scientific journals, including Nature, PNAS, as well as popular scientific outlets such as Scientific American. So without further ado, I really want to thank Rebecca for being here, and we're really excited to hear about her groundbreaking coral research. And I will pass the virtual mic over to her for her to begin her seminar.
All right, thank you so much, Phil. And thank you to everyone at Carnegie for the invitation to speak with you today, and to all of you for joining me. I hope that you are all safe and well at home. Before we get started, I do just want to acknowledge that I've had a few connectivity issues the last couple of days because I'm in Northern California and we're surrounded by wildfires. So if for any reason I do cut out, Phil promised me yesterday that he would tell you jokes until I was able to reconnect. So some of you may be crossing your fingers that actually happens at this point. So anyways, what I wanted to talk to you today about is why coral reefs are important ecosystems, what they offer us in terms of ecosystem services and biodiversity, some of the challenges that they're currently facing. And then I'm going to give an overview about some of the more creative solutions that are being explored right now in order to address the coral reef crisis. And then zero in on the California Academy of Sciences Hope for Reefs initiative in my lab and our particular contribution to this overall story. So when most of you think of coral reefs, you might picture something like this. So they are arguably one of the most iconic ecosystems on the planet, teeming with life and biodiversity, full of color, just incredibly beautiful. You might think of a few charismatic megafauna, a green sea turtle, or a black-fin reef shark, a blacktip reef shark, or an anemone fish or clownfish. Or you may just think of a really nice tropical beach vacation. I know that I would rather be there right now than in my house for the X 100th day since COVID started. But the point being that in developed countries, we largely appreciate these ecosystems for their aesthetics, but it's also incredibly important to understand and appreciate that they offer us unparalleled ecosystem services every single day. So reef building corals play a pivotal role in marine ecosystems. They represent the world's most important biogenic structures. Reefs cover less than 1% of the ocean floor, but they provide habitat and trophic structure for about 1/3 of all marine organisms. And they have biodiversity that really rivals tropical rainforests. You might have heard them referred to as the rainforests of the sea. And in addition to that ecological importance, coral reefs are economically essential for human societies, with more than 600 million people around the world depending directly on coral reefs for their survival. So almost 200 million people depend on coral reefs to protect them from storm surge and waves. And it's estimated that as little as 5 square meters of intact coral reef can mitigate up to 97% of wave energy. So you might imagine a tropical storm or a cyclone that's building intensity as it travels across the ocean basin. And then as it approaches the coastline, you have this huge biogenic structure that rises up from the ocean floor. And that storm's going to slam into the reef before it hits the coastline and mitigate 97% of that wave energy. So in 2004, when we had the tsunami in the Indian Ocean, there were some coastlines that were spared further damage because they had healthy and intact reefs. And in other areas where coral and sand had been mined away, it can cost upwards of $10 million per kilometer to build a wall to protect the coastline. So at some level, this really does boil down to a human security issue, which is why in a lot of areas that are adjacent to coral reefs, insurance and reinsurance companies are starting to engage. Reef fish and other critters are a significant source of protein for up to 1 billion people, especially those who live near coral reefs. And they also offer us tourism revenue, upwards of $36 and $40 billion every year, from people who like to go and enjoy surfing or snorkeling, scuba diving on these beautiful and incredible marine ecosystems. And if you wrap up all of those goods and services in one number, it's estimated that coral reefs provide us free of charge around 375, almost $400 billion worth of goods and services every year. So before we move forward, there's a couple, I just want to do a little Coral Biology 101 really briefly to introduce a couple of little concepts that I'm going to refer to throughout the rest of the talk. So first and most importantly, is a coral an animal, a plant, or a rock? And the answer to that is, it is all 3 of those things. Yes. So if we look at a coral close up, this is a coral of a species called Seriatopora hystrix. It's a bird's nest coral. And what you see here is individual branches. On every single one of those branches, there are tiny little white dots. And those dots are thousands to millions of tiny little coral polyps that are all living in association with one another. So corals are clonal organisms. They consist of thousands to millions of tiny little polyps that are asexually budded from one another throughout the course, the life cycle of this organism. They share resources, they share food, they communicate with one another during critical life events like reproduction. It's an incredibly sophisticated colonial organism. And if you look at one single polyp and break it down schematically, what you'll see is that it looks like a tiny little sea anemone. So it has a mouth. It has a gut. The mouth is surrounded by a ring of tentacles, very much alive. Those tentacles can grab plankton out of the water column and pull it into the mouth and actually digest it, share those resources with adjacent polyps. And underneath all that animal tissue, there's a calcium carbonate skeleton that's secreted by the animal for structure and support, much like you have a skeleton underneath all of your tissue. And so when we refer to corals in the scientific community, we often call them a holobiont because it really refers to this idea that they are a consortium of organisms. There's the actual coral animal tissue, and then this is fascinating. Inside of the tissue lives a often obligatory symbiotic single-celled alga that you will see on the right side of this picture. So that polyp, you see the translucent animal tissue, and then you see concentrated at the tips of the tentacles, all these little brown dots, which are single-celled algae that are critical to the animal's survival. So when you think of a coral reef, you often think of crystal clear waters, tropical locations. You can see for hundreds of feet in these beautiful clear waters. The reason you can see for so far is often because they're incredibly nutrient poor. And so what that means is that there's not a lot of food sometimes for organisms like corals. So in some cases, this algae, which photosynthesizes inside of the animal tissue, will translocate those fixed carbons to the animal and can provide the animal with upwards of 90% of its energetic needs on a daily basis. And then in return, the animal metabolizes and relocates nitrogen and phosphorus and carbon dioxide back to the algae for growth and photosynthesis. So it's this incredible symbiotic obligatory relationship that has evolved over evolutionary timescales. And the reason I'm spending time to explain it is that it's the breakdown of this symbiotic relationship that is causing coral bleaching, which is causing devastation of coral reefs worldwide right now. So most corals start their life as a tiny little sesame seed sized larva, like pictured here in this little video. And they look very simple. I'm going to play it again. They look incredibly simple, but they're actually incredibly sophisticated. So they're sneaky. They have an oral and an aboral end on this larva, and the aboral end is packed with sensory cells, photoreceptors, chemoreceptors, because what that aboral end does is sense its environment. When it's swimming around in its very early days to weeks of life, it probes in its environment to try to find a perfect place to set up life, because once it actually fixes itself and metamorphoses and starts to secrete calcium carbonate and fix itself to the ocean floor, that's a very deterministic decision. That thing is probably not moving for decades to centuries. So these larvae have very sophisticated mechanisms to be able to sense where might be a good place to set up shop in terms of chemoreception and photoreception. So once that larva actually settles and metamorphoses, the aboral end will fixed to the substrate, and then it starts to flatten out, and the oral end differentiates into tentacles. And so you see that here. This is probably around 24 to 48 hours old, 2 adjacently settled coral polyps right there that haven't started secreting calcium carbonate, so all you see is translucent animal tissue. Over the course of the next several weeks, days to weeks, it will start to secrete a calcium carbonate tissue underneath, skeleton underneath that tissue. You can start to see that beautiful architecture of calcium carbonate underneath the animal tissue there. That one primary polyp will then start to asexually bud new mouths and divide. And so what you see here is a single primary polyp in the middle that has now asexually budded 5 new mouths outside of it. This is just a lateral view. This will continue to asexually divide over the course of months, and then eventually over the course of months to years, grow up into an actual coral colony. Fast forward decades to centuries, and you have hundreds of colonies of different species that make up a reef. And then over geologic timescales, you have these huge biogenic structures that rise up from the ocean floor and can be seen from outer space, like the Great Barrier Reef, and that stretches over 2,300 kilometers long. And so the reason that I spend time on this is that I've been studying coral reefs for almost 20 years, and the fact that these huge biogenic structures that you can see from outer space mostly started life as sesame seed sized larvae is something that still just is astounding to me, and I think is incredible. And unfortunately, we're seeing unprecedented rates of loss across these ecosystems worldwide. So it's been estimated that we've lost about half, 50% of corals over the last 3 decades. And we're risking losing around 90% to 99% by the middle of this century. So there was a United Nations report, a special report of the IPCC that was released last fall that looked at the consequences for many ecosystems, coral reefs being one of the most vulnerable, if we are able to cap global warming at 1.5 degrees Celsius versus edging towards 2 degrees Celsius. And what they estimate is that at 1.5 degrees Celsius, if we can cap it at that, we risk losing 70 to 90% of coral reefs. And if we start to creep towards 2 degrees, we're looking at losing around 99% or effectively everything. So this is for a variety of different reasons. We're going to focus mostly on global warming and coral bleaching at the start of this talk, but their local stressors have been affecting coral reefs for decades. So we have a variety of different local stressors. Here you see development. So this is an airstrip that is built directly on top of a coral reef for tourism purposes. You have fishing practices, both subsistence fishing and commercial fishing that's done in a variety of different ways, some of them very harmful like dynamite fishing or unsustainable commercial extraction practices that put undue pressure on coral reefs and trophic processes. And you have pollution that directly, and runoff that directly affects coral reefs. So we have a team of researchers led by Luis Rocha, who's an ichthyologist at Cal Academy, and he is a mesophotic diver, a twilight diver. He does rebreather diving down to 150 meters or 500 feet, hundreds of miles off the coast of continents like South America. And at 500 feet depth, hundreds of miles off the coast of Brazil, he'll still find trash and tires and things like that on these reefs. So there's really no place that we found that they can be removed from these local pressures. But really the elephant in the room right now with respect to impacts on coral reef and how quickly this is affecting and causing kind of catastrophic global decline is global warming. So it's estimated that the world has already warmed 1 degree Celsius or 1.8 degrees Fahrenheit. And so what this is showing, this figure is showing a heating map or a temperature anomaly map from the pre-industrial era or about 1880 to today. And what you can see is that on average across the earth, the oceans have warmed by around 1.8 degrees Fahrenheit. So the oceans cover over 70% of the globe, and they've absorbed over about 93% of the excess heat from greenhouse gas emissions since the 1970s. And so that's offering us an incredible service in terms of climate regulation. The atmosphere would be much warmer if the oceans weren't offering us this service of thermal regulation, but it's not without consequence. We're seeing unprecedented pressures on marine ecosystems, and you've probably seen a lot of pictures like this in the media over the last 3 to 5 years. So previously healthy reefs that are experiencing widespread bleaching and loss. This was a picture that was taken by Luis Rocha and his team just months apart in some reefs in Micronesia that were previously healthy and then experienced widespread global bleaching or widespread bleaching over the time span of just a couple of months. So what is bleaching? Remember that symbiotic relationship we talked about previously. It's a breakdown in that relationship. And so bleaching is a stress response, and it can happen in response to really any stressor. It can happen in response to salinity stress. If there's an anomalous rainfall event, it can happen in response to cold temperature. And it's a stress response that upsets this symbiotic relationship. But in bleaching, isolated patchy bleaching in response to stress, localized stress has been happening probably since corals have been in existence. But what hasn't been happening, the concerning thing, is widespread global mass bleaching, where 70% of reefs worldwide are affected simultaneously. And the only reason that's happening is because of global stressors like coral bleach, like global warming. And that's what's concerning, is the capacity to impact a huge proportion of the world's coral reefs over such a short time period. So we've had 3 global mass bleaching events known on record. So the first one was in 1998. The second one was in 2010, and then the third one started in 2014, 2015, and a lot of people thought it was just going to last a year, and it just kept going and going and going. This is a heat map from that third global mass bleaching event, showing that over 70% of the reefs worldwide were affected by that event that lasted approximately from 2014 through early 2017. And earlier this year, there were reports in the media that the Great Barrier Reef was starting to bleach again, suffering its third mass bleaching event in fewer than 5 years. So it's estimated now that these bleaching events are occurring 5 times more frequently than they did in the 80s, and occurring approximately every 6 years instead of every 30 years previously. And as we talked about previously, corals are very slow growing organisms and ecosystems. And so reefs can take, best case scenario, 10 to 20 years to recover from an acute disturbance like this. And so obviously, if you have systems like the Great Barrier Reef that are being impacted 3 times in fewer than 5 years, and you need 10 to 20 years and longer for some of the longer lived species to recover, those ecosystems are never getting the breathing room that they need in order to recover. So what can we do? So there's this quote that necessity is the mother of invention. And here it is by another great philosopher, Calvin and Hobbes. But what I want to talk about today is some of the creative solutions that are being explored in order to try to confront the coral reef crisis. And I think that there has been, the thing that gives me the most hope is there's a tangible and a palpable shift in the way that people are thinking, the way that people are collaborating and coming together as a scientific community to try to address this in the last 3 to 5 years that I haven't seen in the 15 years previous. I've been in this field for about 20 years. And I find that incredibly invigorating and incredibly hopeful. And, you know, there is no singular solution to this. We're going to talk today about a lot of different things that are being explored. We haven't found a solution yet. There's not going to be a singular one, but there are a lot of really creative things that are being talked about. And so that's what I want to focus on for the rest of this talk. So convening, as an example. So a couple of years ago in 2018 was the first ever Reef Futures Conference. And so this is an effort organized by the Coral Restoration Consortium, which is a group of people who came together recognizing the need for a global coordinated effort to advance restoration science. And so a couple of years ago in Key Largo was the first ever international convening that was a solutions-focused symposium. And the next one is happening next spring, hopefully in person, but we'll see what COVID has in mind in May, I believe, again in Key Largo. The National Academy of Sciences wrote a consensus study report looking at interventions to increase the persistence and resilience of coral reefs. And you can see by looking at the summary or the table of contents that it's exploring all sorts of things here, from CRISPR and Cas9, supercorals to geoengineering and cloud brightening. There is phage therapy, shading of coral reefs, all sorts of things are on the table in terms of exploring how to help confront some of these bleaching threats to coral reefs. And then recently the Australian government announced a new reef restoration and adaptation program, which pledges $150 million to drive innovations in enhanced reef resilience. So there are really 43 concepts that are backed under this plan, from cloud brightening to using concrete blocks to stabilize damaged reefs, to helping corals adapt to warmer temperatures and moving larvae around reefs en masse. So I want to just take a few of these that have gotten a bit more attention than others and talk specifically about what they are and how they might be approached. So one that has received quite a bit of attention is this concept of making supercorals. And so what is a supercoral? What we mean by supercoral is either identifying corals that exist in nature, certain genotypes that might be hardier than others, and then selectively cultivating those, or actually manipulating a coral, certain parts of the holobiont, to generate new assemblages that are more thermally resistant and tolerant to stress. So how would you do this? Remembering from earlier in the talk that this coral really is an idea of a holobiont. It's a complex partnership. We're referring to an entire mesocosm, the actual animal, the algae that lives inside the animal, and then the bacterial community and viral community that coats and is associated with the animal. And scientists are working to alter and tinker with each different part of this with the hopes that it'll enhance the entire system's heat tolerance. So one way you might do this is through creating hybrids or selective breeding. So there's a concept called hybrid vigor, that the hybrid of two individual species might be more tolerant to stress than either of the individual parent species. So some scientists are looking at actually generating hybrids to see whether they're more thermally tolerant. Researchers are also rearing corals in hotter water to see whether offspring inherit adaptations and manipulate genes for heat tolerance. Certain researchers are looking at manipulating the microbiome to help corals resist bleaching and other stressors. And so they're evolving bacterial mixtures or taking corals and moving them from cooler water to warmer water to see how that shifts the microbial association or the microbial community that's associated with the coral. And if it infers a physiological or phenotypic like a preferred trait, some sort of thermal tolerance that they can then preferentially cultivate or breed. And other researchers are looking at adapting the algae. So remembering that the animal, the coral, is typically very slow growing. So in a best case scenario with a fast growing species, it might be 3 to 4 years before a baby coral grows up to be sexually viable and reproductive, and then is actually able to generate a new generation. So enacting evolutionary changes or adaptation potential with a slow-growing organism is very challenging, particularly when we're experiencing the rates of loss in coral reef ecosystems that we are. But the algae have much shorter generation times, on the order of days to weeks. And so there are researchers that are evolving algae for thermal tolerance in the lab, with the idea that you might then be able to inoculate corals with these more thermally tolerant strains and infer thermal tolerance to the animal as a whole. And then we also have efforts, so this is research that was led again by Phil Clevis, who introduced me at the beginning, one of the newest faculty member at the Department of Embryology. And he led an effort with his team to actually was successful in genome editing using CRISPR and Cas9 in a coral known as Acropora millipora. So they did microinjections on coral, on fertilized eggs, and were actually able to induce mutations in targeted genes. And so for this particular application, it was a fluorescence. But the idea here is that you might be able to use CRISPR or gene editing in larvae or adults to identify genes that are critical to bleaching pathways or thermal tolerance, to be able to understand these mechanisms a bit more in depth. Also among potential genetic and reproductive solutions is coral cryopreservation, so building repositories to store material. So cryopreservation is the process of cooling and storing cells or tissues at very low temperatures to maintain their viability. And then you use those frozen tissues to revive coral genotypes that are extinct in the wild, or to use frozen gametes to generate new genotypes. And this is largely through the effort of Dr. Mary Hagedorn from the Smithsonian, who is pictured here, and her collaborators. And she has pioneered cryopreservation of coral sperm, is now possible. And it's even been used in some pilot assisted gene flow experiments. And they have over 50 species that have been collected in 7 different areas all over the world. And US and Australia share a database of about 37 species right now. And so the Coral Restoration Consortium recently formed a cryopreservation working group with the hopes of advancing this critical science. So Mary's group has some early success in cryopreserving and laser rewarming coral larvae. And we want to advance that science and also advance successful cryopreservation of coral eggs and possibly even whole coral fragments, and then train everyone in these standardized techniques. A couple of final things that have been, so we're really just scraping the surface, but there's a reef sun shield that's been made from an ultra thin surface film that shows promise as in the solution for protecting coral reefs from incident light levels over the Great Barrier Reef. So the idea is it's a polymer that would be sprayed across the surface of the reef over larger regional scales that reduces incident light levels by, in early studies, they showed around 30%, reducing the bleaching risk to corals. That's some of my earlier ocean acidification work that I'm gonna skip over today, but there's larval robots that are being led by an effort by Dr. Pete Harrison out of Australia, with the idea that you use these underwater robots to distribute coral larvae in mass and reseed certain areas that were previously depauperate. So really harnessing technology. And then this is a Fulbright scholar that was working at Cal Academy with our Hope for Reefs team in my lab over the last year that I'm going to talk about in just a second, on a project called Coral Maker. That's really exciting. And even earlier this year, there was a new study about bionic corals. And so this team 3D printed bionic corals that were capable of growing microalgae and really defined a class of bionic materials that was capable of interacting with live organisms, with the idea that this would be a tool for advancing animal algal symbiosis and bleaching research to understand photophysiology and stress responses. So all sorts of things are being explored right now. So for the rest of the talk, I want to talk specifically about our Hope for Reefs initiative at Cal Academy and my lab, and in our particular approach and where we're trying to contribute to this. Mostly what we are doing is focusing on scaling restoration efforts, both restoration efforts that focus on asexual reproduction and sexual reproduction, which I'll explain more in a moment. And then I have partnered with the Steinhardt Aquarium at Cal Academy to move forward a conservation breeding program and captive breeding program at our public aquarium. So I'm going to talk about that and how it's a really exciting effort to advance those conservation and fundamental science. So I got really, really interested in coral reproduction when I started grad school back in the early 2000s, because there was this really important paper called Gardner et al. 2003. I started grad school in 2005. And what that paper showed, I did my PhD in Florida, and it was a paper that talked about, for the first time, that there had been about a 50% reduction in coral cover throughout the Caribbean over the last 30 decades. Very heavily cited paper at the time that kind of showed the extent, how extreme this loss was. And so I came into grad school and this paper was being talked about all over the place, and I became so interested and intrigued by the next generation of corals. How were these corals that we're losing going to be replaced? What was reproduction going to look like? Was it going to be able to be sustained under these changing conditions? And that question is what continues to fuel my passion for conservation to this day. So corals reproduce two main ways. So they reproduce asexually, which we talked about a little bit earlier, through budding and fragmentation. So much like a starfish, if you break off the arm of if you break off the branch of a coral, that original coral colony will regrow a branch, and then that branch that you broke off can regrow another part of a coral colony. And then they also reproduce sexually, which is the way that we get genetic diversity out onto a reef. And most coral species reproduce sexually through a process called spawning, where they release gametes into the water column, eggs and sperm into the water column. There's external fertilization that create those tiny little larvae that we saw earlier in this talk, that then swim down, metamorphose, and start to asexually bud and grow corals. So the majority of reef restoration approaches take advantage or exploit corals' asexual reproduction technique. So reef restoration is a practice that's been around for about 30 years. It borrows techniques from silviculture and land-based reforestation efforts. And the idea here is that you would, again, exploit this asexual reproduction technique that corals have to break off and fragment pieces of coral and then preferentially continue to grow them. So this is typically done in nearshore environments or nurseries or land-based nurseries. Here you have some what they call coral trees that are growing. Typically you keep one genotype on a single tree, different genotypes on different trees, different species. And then once they grow to a certain critical size, teams of divers take those individuals and go and epoxy or outplant them back onto the reef. because corals are slow growing, there have been some advances in recent years to try to accelerate this process. So some research that was led out of Mote Marine Lab really identified a process called microfragmenting, where instead of fragmenting entire branches off of corals, you can microfragment smaller pieces. And due to a process that we don't fully understand at this point, it induces some sort of natural healing response where the coral actually starts to grow at sometimes up to 50 times faster than it would normally. So you're actually generating pieces of coral larger than you would under a normal time frame. And because corals are clonal, if you put the same gene, if you keep track of these tiny little fragments and then place them all back near one another, as they grow and touch one another, they will actually, much like skin grafting, just refuse. So you can grow corals that are larger and possibly reproductive size in a much shorter time frame. So one of the biggest hurdles with restoration and the biggest bottlenecks is doing this at scale. So we are very good at this point at restoring coral reefs on the scale of about 1 hectare. But as you saw previously in this talk, when you have an event that impacts 70% of world reefs globally, worldwide, in the time span of less than a year, restoring reefs on the scale of 1 hectare is not quite efficient. And so one of the biggest areas of focus right now at the Academy and at numerous other organizations is how to make restoration techniques scalable with the pace of global loss. And one of the reasons that it is only effective at local scales goes back to the idea that at the end of the day, what you're dependent on is teams of divers going back out and physically putting a coral back out onto the reef, either using epoxy or cement. And that's an incredibly time and labor intensive process. So I'm going to talk about a couple of the ways that we're looking at scaling up these solutions. So one is a concept known as Coral Maker that was founded by Dr. Taryn Foster from Australia. So she spent the last year at the Academy in my lab, with my lab, advancing this idea in partnership with Autodesk, Fulbright, and Cal Academy. And her idea is to integrate robotics and automation into this process to make this scalable. So her idea is that we would use 3D printing to pre-print the coral skeleton so that it doesn't have to spend years to a decade building up that calcium carbonate structure. So you pre-print this coral skeleton and then you pre-drill holes into it that little microfrags could be placed in using robotic arms. So all of this is designed with robots in mind, which is where Autodesk and their pro bono team was completely indispensable. And then you would use robotics to insert microfrags in these that would then start to grow over and fuse and graft the entire coral, and then that coral would be transported back out onto the reef. So you're getting, again, potentially reproductively viable corals at size in a much, much shorter time frame than you would if you had to actually have an individual tiny primary polyp grow up over the course of 10 to 20 years. Another project that we're leading looks at, so that previous project harnesses mostly asexual fragmentation again, but we're also really, really interested in enhancing genetic diversity on the reef. So there are a lot of approaches right now about trying to, again, select these super corals, select which corals might be more thermally tolerant, and then preferentially cultivate those. But there's also an idea that you can just enhance sexual reproduction in corals and genetic diversity that would happen normally. So if we can try to shepherd corals through that sexual reproductive cycle, we get natural genetic diversity on the reef as much as possible, and then nature selects which ones are most fit for that particular time and place. So most corals, about 75% of coral species, reproduce sexually through annual spawning events. So this happens usually once a year, late summer, after a full moon, few days after a full moon, depends on the species, and a few hours after sunset. I always say corals are surprisingly romantic. And so based on decades of research around which species spawn at which times, there are teams of researchers, and this is a project that we're doing in association with SeaCor International, which is a nonprofit that stands for Sexual Coral Reproduction. And these teams of researchers go out, and we know when these corals are likely to spawn. We net the coral colonies, and these gamete bundles that are released that contain eggs and sperm are very lipid heavy. So they float when they come out. And so they rise up into the net, into these little collector cups at the top of the coral. And then you can actually screw those off, cap them, and take them back to the lab in order to fertilize them under optimal conditions. If they're released into the wild, a lot of times it's a feeding frenzy. Fish love to eat them. You lose up to 99% of this stuff just from predation. So what we're really trying to do is collect as much as possible, take it back, shepherd it through these really vulnerable early life history stages under optimal conditions, and create as many babies as possible that we can then translocate back out to the reef. So these are eggs, and the water in each of those tubes is cloudy because the sperm dissociates into the water. So each one of those gamete bundles has a sperm packet in the middle, and then it's surrounded by eggs on the outside. And they rise up to the surface of the ocean, and then after a certain amount of time, the sperm packet will burst, and it causes the eggs to it causes the whole packet to dissociate, and then everything mixes with other genotypes, and they fertilize in the water column. Here's a close-up of some of the coral eggs. And then this is us cross-fertilizing, cleaning eggs and cross-fertilizing everything. So we're mixing millions and millions of eggs together, different genotypes together, to try to maximize genetic diversity. And then what we do, once those larvae, we have settlement substrates that we've conditioned. And then once those larvae have developed, depending on the species, that can be anywhere from 5 to 10 days after spawning and fertilization, we settle those larvae onto these little substrates. And you can see here a tiny little coral juvenile. And then those substrates are then planted back out onto the reef. And so this process, Sea-Corps has been doing this process, a lot of researchers have been doing this process for quite a long time now, around 15 years. And this is a paper that was released several years ago that showed that one of some of the Acropora colonies that had been reared in the lab and planted back out actually grew up and spawned with their native populations, which was really exciting. So the bottleneck with this approach is similar to the bottleneck we talked about previously, which is the bottleneck of actually having people go out and physically plant these things onto the reef. Incredibly time and labor intensive. And so one of the potential solutions that we've been looking at is a partnership between the Nature Conservancy, Sea-Corps International, Autodesk, and Cal Academy is creating self-stabilizing substrates that these larvae can be settled onto. And then instead of actually going out, previously larvae were settled onto terracotta tiles that had to be epoxied or fixed, like nailed into the reef. But if you settle them onto these self-stabilizing tetrapod-like substrates, then the idea is that you could scatter these out on the reef with much less effort in terms of diver time. And there have been preliminary studies that show that these things are actually move fewer than 50 centimeters over the course of 6 months, and gives the coral enough time to grow up and actually solidify itself to the reef. So here's a picture on the left. You have a couple of tiny little coral recruits as that is being put out onto the reef. And then on the right, you can see that these corals have actually grown up and started to adhere to the reef. So the idea here again is doing this in mass. You have hundreds to thousands of these things that are conditioned, hundreds of thousands, millions of larvae that are settled onto these, and then eventually working towards the idea that you'd be able to scatter these around and really scale up the amount of coral juveniles that are being newly sexual recruits that are offering genetic diversity that are being put back out onto the reef. And the last thing that I want to talk about today is an effort that I started a few years ago at Cal Academy in partnership with Steinhardt Aquarium, which, excuse me, is focused on spawning corals in captivity or in land-based conservation breeding programs. And so this is a concept that was originally pioneered by Jamie Craggs and his team Project Coral out of the Horniman Museum in London. And then we started a project 3 years ago called the Coral Spawning Lab at Cal Academy, and we were the first group in the US that we know of that was able to spawn corals in captivity. And there are also efforts that are starting at public aquaria all around the world at this point, which is really exciting. And so the goal here is to develop protocols for predictable broadcast coral spawning in captivity, much akin to how you have conservation breeding programs for all sorts of endangered animals in zoos and aquariums all over the world. And so this may sound a little bit easier than it actually is. Anybody who has tried to keep corals happy and healthy in aquaria, it is not a trivial task. They are notoriously fickle animals. And so that's where this partnership with Steinhardt Aquaria really comes into play, is that it's such a strength to be able to partner with aquarists and hobbyists that know how to keep these animals happy and alive. So it's generally accepted that what causes corals to spawn in late summer, days after a full moon, hours after sunset, is this hierarchy of environmental cues linked to seasonal, lunar, and daily environmental rhythms that work over finer time scales to actually determine the exact time of spawning. There's a lot of different things that probably play into it: nutrition, seasonal temperature, solar irradiance, photoperiod. We're manipulating 4 of these environmental cues in our system that have shown to be successful for the species that we have started working with, which is Acropora hyacinthus. And so what we've done is mimic for the corals were collected from Palau. These corals were collected from Palau. And so we have sophisticated software and sensors and lighting regimes that are able to mimic seasonal changes in temperature. So we plug in, we have 10 years of historical sea surface temperature data from Palau, from the government, that we are mimicking the monthly mean temperatures for every month of the year, and then iteratively every single day, our software is actually changing the temperature incrementally every day to increase or decrease towards the next month's average temperature, so that these animals experience a winter and a summer. We are mimicking lunar cycles. So these animals have to experience a full moon and a new moon. And so we have the day of the new moon programmed into our system and the day of the full moon programmed into our system. And we have LEDs that are changing intensity ranging from 0 to 100% every single day throughout the course of the month. We have to mimic day length. So we're changing the sunrise time and the sunset time. We have our lights. We have 8 different LEDs and wavelength, or we have LED lights that have 8 different wavelengths that during sunrise, which we ramp up the intensity over 3 hours, and the not just the intensity, but the distribution of wavelengths is changing over the course of the day to mimic different times of day. You might get more white light at peak noontime irradiance. And then we also changed the solar insolation based off of historical data that we've pulled from government websites. So we have about a 22-year solar insolation data that we change over the course of the year in order to mimic those patterns as well. So I want to show a very brief video of this effort and kind of how it started and progressed over the last few years. We just celebrated this spring our 3rd annual spawning event with this species. And so again, this is in the middle of Golden Gate Park in San Francisco, so we're not adjacent to a coral reef, which makes it tricky. And because we didn't want light pollution, we had to build a room inside a room, which is what that was showing. So it's akin to a photography dark room. So we sent a team of researchers to Palau to collect coral colonies. We did this sustainably. We never took an entire genotype off of a reef, no more than 1/3 of a coral colony. And we've gone back and checked the colonies we've collected and made sure that we're not having harmful impact on the ecosystem. These colonies were shipped overnight back to the California Academy of Sciences. This is about 36 hours in transit back to the lab. Again, this is where partnering with a professional aquarium is incredibly invaluable. They are masters at knowing how to transport organisms and keep them healthy. And then we mimicked all of these different environmental cues, and we were actually able to elicit synchronous spawning, which is really exciting. So our first year we had, I believe, 7 animals that spawned synchronously in our system. And we were able to cross-fertilize all those and generate larvae that we then settled and reared out over the last several years. We have a lot of different scientific projects associated with this, where we have collaborations, which I'll talk about next, with Stanford and Berkeley, NASA Ames, all sorts of different scientific institutions in the area, in order to not just use this as an applied conservation technique, but also really just advance fundamental coral biology and science. So these are the coral larvae that are being reared over the course of 5 to 7 days. They start swimming around and searching for an appropriate place to settle. Here are some of our preconditioned settlement substrates, and the larvae swimming down to explore and see if they want to settle. This is a newly settled coral baby, and then sometimes you'll get gregarious settlement where they all just settle on top of one another. These are hundreds of individuals that have all settled on top of one another. We co-culture with urchins in order to clean off algae and things that might compete with the corals, and we also inoculate them with symbiotic algae early in the process to enhance growth and survivorship. And so there's so much that you can do with a system like this. I'm just going to let this video run its course. It's just a few more minutes. I don't want to miss the acknowledgements. There's so many people, so much support that was critical to this. Okay. So here are these are babies from the first year that we did this. And so, or these are babies, sorry, from 2018, actually. So these are 1.5 years old. Actually, when I took this photo, they were about 1 year old. So these are a copper hyacinthus, 1 year old, starting to branch. They, I haven't been back as much as I had hoped over the last 7 months just because of COVID, but they are even bigger, even more beautiful now. So we have one cohort from 2018, and then we have from this spring another cohort now that I don't have a picture of, but they are about 6 months old at this point. So we have multiple years of cohorts. And this year, which was really exciting, was the first year that we got respawning of individuals. And so what you see here is a branch of a coral colony that's, that I purposefully broke off to look for, to see whether it was gravid, which just means reproductively viable. So I'm looking to see if they have mature eggs and whether that colony might spawn. And so this was this picture. I took this picture in April, and then these individuals spawned in late April and again in May. And here's another picture from a different colony. And so these were individuals that spawned last year. We collected last year, spawned last year, underwent the entire gametogenic cycle in our system, which in this species we think is between 7 and 9 months, and then spawned again, which is really exciting. So the goal is to keep them reproductively viable year after year after year. So now we are looking at adding new species, and really now that we have proof of concept, just kind of, there's so much you can do with a system like this. So all of the different things that we can do from an applied research and basic research standpoint. So some of the things we have going right now are just contributing to this optimizing land-based conservation breeding protocols concept, which is another charge of the Coral Restoration Consortium. I hope to use this system to be able to contribute to advancing the cryopreservation work that I spoke about earlier through, again, the Coral Restoration Consortium. I have a student that is looking at carryover effects to optimize growth and survivorship in early corals. So this is another huge bottleneck, is that we know very well at this point, usually how to get 90 to 100% fertilization success, 90 to 100% settlement in coral larvae. But then, looks like something just happened on my end. Let me just make sure that. I think I'm still going.
You're good.
Okay, thanks. But once you plant those individuals back out onto the reef, you usually have very low rates of post-settlement survivorship, a lot of times just because you're planting them back out into the environment that was degraded in the first place. And so what we're trying to do is explore positive carryover effects. There's quite a large body of literature about negative carryover effects, that if you this is true for all sorts of different animals, that if you expose either the parent or the offspring in early life stages to a stressor, that will implicate fitness and survivorship and growth later in life. But turning that on its head to explore positive carryover effects is something that is much less explored. So this student is looking at whether there are low cost, easy to implement things that we can do in terms of husbandry techniques, in terms of like sea course techniques, whether we can inoculate earlier with certain strains of algae, whether we can change our feeding regimes, our densities that we're rearing things out, whether we can do hardening techniques of the seawater to boost calcification early on. All of these things, what we want is to identify things that could be implemented at scale and really enhance survivorship. And then we have basic research that we're doing. So in association with, or collaboration with Stanford and Steve Palumbi's lab, we are currently in the genome assembly stage for this species with dovetail. So we're generating a new genome that would facilitate all sorts of different kinds of research. We have a collaboration going with a PhD student, Alora Lopez, again, out of Steve Palumbi's lab at Stanford, who is looking at, might be interesting to the embryology contingent today. We don't really have strong evidence one way or the other whether corals have segregated germlines, which means that at the early in life, and a lot of animals, you will have, your cells will either differentiate into germ cells that produce eggs and sperm, or they will segregate into cells that then undergo somatic mutations or will generate different somatic cells throughout life. We don't know whether that is true for corals. And so one way that we tried to address this last year, we did this study, was by looking at somatic mutation rates throughout an individual coral colony. And we did this for several colonies that were in our system and look at whether there were mutations. We were able to take individual branches off of the coral colony from different areas on the colony. And then when that branch spawned, we were able to collect the tissue, the adult tissue from that branch, and the sperm cells from that branch, and see whether there were mutations present in the sperm that weren't present in the adult, or vice versa. So if there were mutations present in the adult that weren't carried over into the sperm, that would be evidence that they have segregated germlines, and that the sperm don't have the ability to propagate these somatic mutations. And this is the reason this is important, is it's incredibly important in terms of adaptation potential and evolutionary potential for corals. So some of these very fundamental things aren't known for corals because they have never really made a very good model system because they're hard to keep in captivity. And so having access to these animals throughout their whole reproductive cycle opens up just a Pandora's box of things that we can do. We have a collaboration with a researcher named Dr. Lishko from Berkeley, looking at an ion channel that's critical to sperm activation called CatSpur. And how sensitive that ion channel may or may not be to changes in water chemistry. And I'm hoping to start a project this year looking at microbiome changes throughout the course of the gametogenic cycle, and whether there might be certain microbiome changes that happen around the time of spawning that are actually important in triggering a coordinated spawning response. There's been, again, we don't really know how corals spawn synchronously. There's been a little bit of work showing that there might be pheromones that actually trigger corals in adjacent areas to spawn at the same time. What I'm kind of interested in knowing is whether there are shifts in the microbial community that might implicate quorum sensing that might be playing a critical role in the synchronized spawning result. And that's another huge benefit of having access to the same coral throughout its entire reproductive cycle. So all sorts of things you can do. And I would really love to speak with some of the embryology faculty and students online today if you're interested in exploring any of those. So just wrapping up some of the, the idea here, now that we have worked out, and not just us, this again was pioneered by researchers in Horniman, but now that there's a group of people at different institutions that have worked out captive breeding of these animals, and we know we can shift it. So in our case, we are spawning corals. This species of corals usually spawns at around 9 p.m. at night. That means if we want to do science, we have to be up until 3, 4, 5 o'clock in the morning. I've done that for 5 to 10 years, and it is not pleasant. And so what we've done is actually shift them and trick them into thinking that we simulate sunset at about 9 o'clock in the morning. And so they spawn at 1 in the afternoon, and we have all day to do our research. So we can play with some of the cues that we're giving them since we're simulating them. So one of the ways you could do this is actually shift the seasons and get multiple systems from multiple with different seasons, and then have staggered spawning throughout the year. And so one of the hurdles to advancing coral science has been this fact that they typically spawn once a year. You have one chance for scientific output and productivity, and if you miss that window, you have to wait another year to advance it, as opposed to other model organisms where you have multiple opportunities every week, every month in order to conduct science. So if you have multiple systems that are staggered, you have more opportunities to advance scientific output on a faster time scale. And then we've also done some just really fun, being based in a public science museum, we've done some really fun outreach efforts. So this year we set up, because of COVID, a coral spawning camera so that people could sign on and see coral spawning live. So here are a couple pictures of the red lights when it was nighttime, and then the camera when it was during the day. And I did a live stream one day. So up here, I don't know if you can see my mouse, but up there I'm pointing to a coral that's actively spawning. And I just talked to the public about what spawning was and what they were seeing. This event once a year for about 15 minutes is how much it lasts. So some really fun education and outreach events with this system too. So anyways, I'm going to end it there and leave plenty of time for questions, but I do hope that, none of this is to try to instill a sense of false hope. It's incredibly important that we all understand the gravity of the situation with coral reefs and what they're facing right now. But I also think that it's important to tell all of the hard work stories that are happening and all of the creative solutions that people are exploring and just pouring their hearts into every day. And so my hope is that today has both instilled you with a sense of the gravity of the situation, but also left you with a sense of energy and hope around the prospect that we may be able to come together and move forward some interdisciplinary work that actually moves the needle in the right direction. So thank you. And with that, I'm happy to take any questions.